100 people signed up, and there are now 51 in the meeting. So I think we'll wait a few minutes before I start the introduction. Den gar nicht weit weg. Okay, I think we'll start now. So again, welcome everyone to this CBIO, the Center for Circular Bioeconomy at Aarhus University, our webinar sessions. So this webinar is about green biorefinery and the results from feeding trials with the green protein, also known as leaf protein concentrates from green biomasses. So my name is Morten Ambye-Jensen. I'm an associate professor at Aarhus University. I'm head of the Center for Circular Bioeconomy, and I will be moderating the webinar today. First off, a few informations and house rules about the online webinar. So it was a very popular topic. We could see we have 100 people signed up. Probably not all will attend, but we are now 65 participants, which is really great, and we're happy to see so many of you, or at least know you're listening in. First off, we are recording the webinar, so you should be aware of that. And this is also it will be put up on our CBIO webpage, so people can see it again or at another time. Please mute your microphone, and during the presentation, so we have 4 different presentations. If you have questions to the presentations, please post it in the chat. So ask all questions in the chat while the presentations are on. Then in between presentations, maybe we have time for 1 or 2 short questions, but otherwise we take the discussion and question session in the end. Yes, so let's go on to the program. So today, as I said, we'll have 4 presentations, starting out with 2 researchers from Aarhus University. and an industry presentation from Vestjyllands Andel, and lastly, turning attention towards Ireland and hear about the experience from there. I'll be introducing each of you before your presentation start. But first off, I'll just like to take the opportunity to tell a little bit about what we do at CBIO and what the Aarhus University Center for Circular Bioeconomy is. So we are a research and innovation hub, a network among researchers, and also reaching out towards industry organizations and policymakers, all kinds of stakeholders that are interested in circular bioeconomy. We are on a mission to solve grand societal challenges regarding food production, materials production, energy, climate, and environment, which is all parts of challenges that we can solve in a holistic approach towards circular bioeconomy. So the center includes 8 different departments at Aarhus University, bridging the and covering the topics of biology, technology, sociology, environment. So we have a focus on circular utilization of bioresources, how we get bioresources into production systems, how we process and produce desirable products that consumers actually would like to have, and how the resources are recycled back. So we see ourselves as the Aarhus University partner and entry point for collaboration on circular bioeconomy. And today we will focus on the green biorefinery, which has been a big part of CBIO's activity over the last 5-6 years since we were established. And green biorefinery is a really good example of a value chain that bridges across many disciplines and are true interdisciplinary in its core, because we need to get everything from the field, from agriculture to connect to processing, logistics, product applications, livestock and animal production, and therefore feed, as well as a lot of other subjects: food, materials, energy, climate, and environment. So with no further ado, I will introduce Lene Stødkilde, colleague and researcher at the Department of Animal and Veterinary Sciences, Aarhus University. And Lene, you will present digestibility of green protein for monogastrics. Lene have been a key researcher in all the feed trials that have been carried out with the green protein concentrates produced here at Aarhus University, both in early pilot plant and now at our demonstration facility in AU Viborg. So please, Lene, I will stop sharing and you can take over the screen.
Yes. So thank you very much, Morten. Can you see my screen in presentation mode? Good.
Yes.
So I have been asked to talk a little bit about the feeding trials we have done at Aarhus University since this green biorefining concept was initiated as a focus research discipline in 2013. Yeah, and I will focus only on the feeding trials for monogastric animals, as Morten just said. Yes. So as you probably all know, we have as a mitigation strategy for these environmental and climate issues that we are facing, we will see a very changed landscape in the future with less annual crops such as maize and grains, and a higher proportion of perennial crops like grasses and legumes. And at the same time, we also face a future where we must have much more sustainable food production. However, we do also see a future where we have a very increased need for especially animal-based protein when we look worldwide, and all these things combined will challenge the agricultural area and limit the available agricultural land that we have, so that we will see less arable land use per person in the future. So this means that we must produce more efficiently, and we must use the resources we have much more efficiently. In Denmark, we also see a clear demand for phasing out this import dependency we have of many feed products, and I know it's the same in many other European countries as well. And all these things combined, they lead to this idea of using the perennial crops as an alternative, more sustainable protein source for our monogastric animals. In this search for an alternative, we must also remember that what we are trying to remove or substitute is primarily soy in a Danish context. And soy is a relatively cheap, available, high-quality protein source, which completely dominates the protein that we put into our monogastric animals. So you could say this search of looking for a more alternative protein source, it's complicated, and if we need to be successful, we have to address this from different angles. We must look at the development of the necessary technology, but we must also look at the yield, meaning how much can we produce from this future limited agricultural land that we have. How available is this new protein source, and importantly also, what is the quality of the protein source? So all these things need to be combined in focused research in order to successfully develop or implement a new protein source. And in this talk, I will primarily talk about the quality of this protein source. But just remember that all other issues are also very important. When I present studies on green protein, I will call it green protein, but it can originate from many different sources. So primarily we use grasses and legumes as input material, but I only talk about the product that is made from macerating the plant material and precipitating the protein in this green juice that we get. And this protein concentrate will be suitable for monogastric animals, so pigs and poultry, humans, but could also be used for fish, for example. The process can be optimized in all steps from field and until the final product. And we can get many different products produced. And in this process, we will also get a residual pulp or press cake, and this juice remaining after we precipitate the proteins, but I will not address them as suitable for monogastric feed purposes in this talk. So only the protein concentrate here. So when we look at the quality and evaluate the quality, we mainly look at these three parameters. So composition, digestibility, and productivity studies. What we see is that we can produce protein concentrates having a crude protein content ranging from 30 to 70% of the dry matter. It will depend on what we put into the biorefinery and how we what kind of parameters we use in the production, but around these numbers. We also have a very interesting amino acid profile, which is comparable to what we see in soy, and we have a high content of essential amino acids, making this protein very interesting. We also have a high fat content. Normally we have 10 to 14% of the dry matter being fat, and it's mainly omega-3 fatty acids that we have in our product. And we also have a high content of fiber, and it's kind of a balance. If you have a high protein content, we will also have a low fiber content, and vice versa. And I will get back to why this can create some challenges. We have done several digestibility trials throughout the years where we have used rats as models. We have done trials with pigs and poultry, and we have also looked at in vitro digestibility. And then finally, the productivity studies have focused on pigs, both organic and conventional pigs, but also layer and broilers. When we started evaluating the quality, we used a rat model because we could produce sufficient amount in the lab to be able to feed these rats. A rat model will also enable us to kind of screen many different products, and it's easier to handle than doing pig models. So this was where we started. And what we could see in these studies was that the protein digestibility is very closely linked to the protein content. So the more protein we have, the higher digestibility. And we believe that this is because of these fiber issues. So if we have a low protein content, we will have high fiber, and then we will probably also have more protein bound to the fiber and being inaccessible for the animals. So what we aim for is a high protein content. We can see here in the circle, these preparations are very early products we made where we had a protein content of 30 to 40%, and we were not able to reach the same quality as we have here in soybean meal. But we also saw that in later preparations, where we could increase the protein concentration, we were actually able to get digestibilities that were higher than the soybean meal and approaching what we see in these high quality protein concentrates from milk and potato. So these rat studies, they indicate that there's a huge potential in the quality or in using green protein as protein source for monogastric animals. Later, we moved from the lab scale into pilot and demonstration scale here at the university, and were able to produce enough protein to do digestibility trials in pigs. And this figure shows some results where we have looked at the ileal digestibility of crude protein and lysine in pure stand preparation, so protein extracted from different legumes and grasses. And what we can see here on the left, where we have protein with a protein content just above 30%, we can see that we get digestibility of crude protein of approximately 60%, which is much lower than what we have in soybean. On the right side, we have some later preparations from red clover, lucerne, and festulolium, where we can see we managed to increase the protein content. And this increased protein content is followed by an increase in the ileal digestibility of both crude protein and lysine. And for if protein extracted from festulolium and lucerne, we're actually relatively close to what we see in soybean meal. But these studies, they just confirm that we need to focus on increasing the protein content of our product, and then we will also hopefully see a higher digestibility. So this is the hypothesis, at least. We have looked at optimizing the biorefining process in several projects with the aim of identifying parameters that would give us a high yield, a high protein content, and hopefully also a high quality. And as a general rule, we can see that the protein digestibility is affected by all choices we make in the process. So everything we do will in the production will affect the quality. Here I present two different trials we have done. On the left, we have a study where we looked at the effect of a harvest method. So whether you chop the grass, here's grass clover mixture, but whether you chop that in the field or you just harvest it as a long plant, mode sample. And then we also look at the effect of postponing the biorefining process, so waiting 6 or 12 hours after the harvest to process. And the reason why we do this is to kind of introduce a buffer when you move into commercial production, where you typically will transport your plant material for longer distances. Then it's important to know how fast you need to process the material in order to get a high quality. And what we can see is that there's an effect of both the harvest method and time on both quality and protein content. And the best results, we will get the best results when we mow the grass clover, so we don't chop the plants in the field, and we process them immediately after harvest. So we don't want to wait too long in order to get a high quality. On the right, we have also looked at the effect of introducing different precipitation methods. Normally, we will precipitate the proteins in the juice by heating them to 85 degrees. But we have tried to see whether we could improve quality by precipitating in a different way. For example, by removing some of the fibers or doing a two-step heat treatment. And what we can see here on the black line is that we can increase the protein content. The first two bars, they represent the standard precipitation. We can increase the protein content using different methods, but we can see that this is at the expense of the quality. So when we further refine our or product or introduce different additional processing steps, we do get a higher protein content, but a lower quality. So we believe that this is due to the extra time that is needed to further refine the product. And it will also allow processes within the plant to modify the proteins, leading to a lower quality. So the overall conclusion from many of these digestibility trials we have done is that we can produce protein of a high quality, but we still have values that are below those of soybean meal, and we see large variations in the quality depending on the choices we make in the process. And it looks like this fast precipitation method of simply heating the juice will result in the best quality. So when we then move to production trials, where we look at how do the pigs actually utilize the protein, we see a slightly different picture from what we saw in these digestibility trials. We have done several larger production trials at the university, or where we have been involved. We have done conventional and organic trials where the design has either been a dose response trial or where we have partly or completely substituted the soy products in our feed. We have looked at the effect of adding green protein, so where the green protein has been the main focus, or we have just used green protein as a local ingredients, but where the focus of the study has been something else. I don't have time to go through all these studies, but I can just give you the general picture or message from all of these studies. And it is that we see the pigs perform very well when we add green protein to the feed. So despite these lower digestibilities we saw in our ileal digestibility studies, we see very nice results in our productivity trials. We see no negative health effects in the pigs. We have the expected growth. The pigs utilize the nutrients very well, and we even see some indications of a higher meat percentage when we add green protein. We can also see that the pigs, they experience increased oxidative stress due to this high content of unsaturated fat that we have, but it seems like a high content of antioxidants in the form of vitamin E will also kind of counterbalance this stress. So although we have a high content of fatty acids or unsaturated fatty acids, we do not see any problems with rancidity or structure of the meat or health problems or anything like that. Yeah, and we have done several sensoric analysis of the meat, and the meat is of good quality, and the shelf life is also fine. And then we have some preliminary results from a study that does not indicate that that we have an increased leaching of nutrients when we add green protein to the feed, and we do not see increased greenhouse gas emissions. So overall, these production trials, they tell us that green protein is a good substitute for soy. We do not see indications of anything different. So I just want to shortly change the animal species. When the first trial we did with animals was done on organic broilers, where we use protein extracted from organic grass clover. We had a protein product with a concentration of protein of 36%, so this was in the lower end of what we normally see now. We did a dose response trial where we had different inclusions of green protein in the feed mixture. And what we could see was that when we added 8% green protein in our feed, we did not see any negative effects. The growth was similar to the control group, but when we add higher amounts, we see a compromised growth and slaughter weight. And what is interesting with these broiler experiments, and also something that is seen in experiments with layers, is that we have a very clear yellow coloring of meat and fat when we include this green protein in the feed. So this is something that we will need to be aware of if we use green protein for broilers. And then finally, we have an ongoing project in a Norwegian collaboration where we have done a feeding trial with broilers using protein that has been produced at the new biorefinery in Steinkjer in Norway. A dose response trial was also done here, and the trial was completed with a nice, it went very well, and we are currently analyzing data and looking at samples. Yes, so the last slide, of course, we have some unanswered questions, which will, of course, be our focus in the future. We have these variations in quality that create some challenges with the current feed practices. So these has to be managed somehow. And then we have this balance between how do we obtain optimal yield to use the resources we have efficiently, how do we get high protein content, and how do we get the best quality. And currently we don't have any solution that would fit all of these parameters. So this is also something that we will work with. And then we have this issue of pigs responding very well in production trials, but we still see this lower digestibility when we do our pig trials. And of course, we would like to know why. And I think in the future, we would look more into digestive mechanisms within the pig, what will happen in the intestine when we feed with green protein. And perhaps there will be some beneficial health effects in the green protein that could also add to the value. So much more work to be done in the future. And of course, all these studies have been supported by many different grants. And yeah, so thank you very much for your attention.
Thank you, Lene. Thank you for the presentation. I'll just share the program again. Yeah, so do you see the program? Yeah. I think we will wait with the questions. Lene, maybe you can have a look in the chat and reply via writing, so, and then we can follow up on some of the issues later. So now we'll turn to the next presentation. So we have Nikolaj Peder Hansen, also from Department of Animal and Veterinary Sciences here at Aarhus University, who's been working hard on the activities regarding the press cake fiber for ruminants. So we'll turn to the cows now. And Nikolaj, you can share your presentation and start when you're ready.
Thank you. I hope you see my presentation here.
Yes, that's perfect.
Yeah, thank you, and thank you for the invitation. And as you say, I would like to present a bit of the results that we have had through the last couple of years, where we have been looking on the value of the pulp for ruminants. And in this context, I always like to start out with showing this table. A few years back, Uffe and company, they did some calculations, and basically what it says here is that they try to calculate the area needed to substitute the imported soy protein, the area needed for grass production, and it's put up like different scenarios. So if we look at today's scenario, you can see that it's about 1 million hectares that they suggested that would be needed to produce green protein. And if you look a bit ahead into the future and into an optimized scenario, then they suggest that it's about 500,000 hectares of grass that was needed. And I'm a dairy nutritionist, so I like to translate a little bit into how many cows do we need then. So the point here is to see how much pulp is produced on these two scenarios, and if I assume that each of the cows that we have eat about 10 kilograms of dry matter per day, then we would need approximately 1.7 million cows, dairy cows, in today's scenario, and 1.3 million cows in a future scenario. And yeah, to put that into perspective, in Denmark we have about 650,000 dairy cows currently. So this slide here is just to illustrate that if we move into a production like this in any way, then there will be plenty of pulp, both especially for feeding the cows and also for other purposes. So it's nice to start out like this. But just like Lene, I'll just give a short introduction to like what is the pulp and how does it fit into a dairy ration or a ruminant ration. And what you see here is the pulp we used in an experiment a few years ago, where you see that the pulp is compared to what we normally feed to the cows, this precision chopped grass, where you see that these fibers are quite disintegrated and the particle structure is much more open. So that's sort of the visual, what you can see, how it looks like. If we dive a little bit more into see what are the macronutrients in the pulp, then what I show here is, if you look on the y-axis, you have the crude protein concentration, and if you look on all the green dots, that's where you have the concentration of crude protein in, you can say, the parental forage, so the forage or the grass clover that you put into the biorefinery. And if you look on the x-axis, you can see what the NDF concentration is, so the fiber concentration. And if you then follow the line to the red dot, then you have the concentration of crude protein and of fiber in the same, you can say, biomass, but as the pulp instead. So all the lines here, they represent different data from different literature, and it basically shows that going from the green dots to the red dots, you generally see a decrease in crude protein concentration, but a huge increase in fiber concentration. And you can see on the solid lines here that that should indicate the biorefining of fresh grass or grass clover, whereas the dotted lines, they represent biorefining of something that has already been ensiled. So it also reflects a little bit how the approach to biorefining is, depending on mainly the countries. So that tells us that basically that NDF fiber concentration increased a lot, and that's also what we see when we run our laboratory analysis on digestibility. We also see that when NDF increase, we see a huge decrease in digestibility. And I think cows are a solution to many things, but in this context, I think that it's obvious that we can take some of these low digestible feedstuffs that has a high fiber concentration, and through the cows convert it into something that is nutritious, like milk and meat. And throughout the years, there has been some research done on this. Just to mention a few things here. Back in 2019, Vini published a paper on a production trial that was performed here in Denmark, meaning that a lot of cows tried to eat rations where they either got this traditionally chopped grass clover silage, and also compared that to other cows that were fed this pulp from grass clover silage. And what we mainly look on is the dry matter intake, where in their study they didn't find any difference, whereas for the milk yield and energy corrected milk yield, they saw about 10% increase in production. So that was quite interesting. And what is specific for this experiment here was that they fed pulp that was produced from fresh grass clover. If we jump in another direction, there is also a study here from Finland, where they, compared to the Danish study, they produced pulp from silage, and where they also fed 3 different treatments to dairy cows. Either a treatment with 0% pulp, meaning that there was a normal grass silage, or they substituted some of the grass silage with either 25% pulp or 50% pulp. And what you see here is that there was only an effect on the 25% pulp, where they ate a little bit more, but otherwise no statistical difference on the milk production, although there was a tendency for a reduced milk production on the 50% pulp. And I could continue. There are some other studies out there also coming from Ireland, but the bottom line is that there is still a very limited number of studies that investigate this, the production response on feeding pulp. And what we generally see is that basically there is a huge variation in the quality of the pulp, because we both see a huge increase in milk production and a decrease in milk production and so on. And some of these things are probably caused by mainly the efficiency during the processing, like how intense is the processing of the input material into the biorefinery. And of course, it could also be some of the interactions between this efficiency during processing and, for example, the developmental stage at harvest. So one of our hypotheses about especially the improved milk production when feeding this pulp is that when we do this biorefining and perform this physical processing of the grass, we see an increase in the surface area of the feedstuff. So on the left side, you see a microscopic picture of a grass particle that has been precision chopped. So that's again how it looks like as we feed it now for the cows normally. And then on the right side, you see a microscopic photo of pulp from the same grass. And you can see here again, like also with this magnification, that the particles has really been disintegrated, and the hypothesis is that the more it's disintegrated, the easier the easier the microbes in the rumen has to access all these particles and starting to degrade the particles, so that the efficiency for degrading is better. That was one of the hypotheses, and this is just to show you the experiment we ran a few years ago, where we want to investigate this. So this was just a traditional in vivo digestibility trial with the 6 Holstein cows, fed 6 different treatments. And the point here is that we made a treatment where, like we do normally, so that means that we mow grass, as you see on the left side of this photo here. We mow the grass, we wilt the grass, we precision chop it, and then we ensile it, and that's sort of the control. And then we wanted to produce some pulp, and we did that by harvesting, direct harvest, as you see on the right side on the photo here. And then we pressed this grass once to get some pulp. And then to sort of mimic a very intensive processing in the biorefinery, we took some of this pulp and pressed it twice. So that's the 3 treatments you see on the bottom here. And then to get a bit more variation into the experiment, we did this on grass that was harvested either on an early maturity state or a late maturity state. And basically, we harvested like with the maxi grasser here, and then it's produced in the demonstration platform here on AU Viborg. And then the pulp was then, and the grass clover, chopped grass clover was then ensiled in these barrels. And that's then fed to cows in a normal ration, you can say, with soybean meal, wheat, and minerals, and 65% of the ration was then one of these silages. That's then tested in our barn, where we have our ruminant cannulated and intestinally cannulated cows housed. And the main things that we wanted to look on here was fiber digestibility and protein value. So if you look on the graph on the left side here, you have again the treatments here: chopped grass silage, pulp pressed once, pulp pressed twice, and then the early and late developmental stage. And what we look on here is the rumen fiber digestibility, and here it shows that for the treatments we had on the early developmental stage, we don't see any differences here, probably because that the digestibility of the grass was already very high, so it's difficult to improve it even more. Whereas if we look on the grass harvested on a late developmental stage, there you see that digestibility starts out being a little bit lower already, and then when we process it with this physical treatment, then we can also improve fiber digestibility. And if we then turn to protein value, you can say that for many traditional Danish rations, we already have a little bit too much rumen degradable protein in our rations. So we really want to move some of that protein degradation from the rumen to the small intestine where it can be absorbed. And that's what the figure on the right side shows here. Basically is that you can say the amino acid digested in the small intestine, which is AAT in the Danish system, is improved when we feed the pulp that has been pressed twice. So, and a last thing from this experiment is that we also measured methane production, and we do that using these respiration chambers here. And what we saw was that statistically there was no difference between the treatments.
Precis. Det var det.
I think what we can see is that there is no difference between the treatments in terms of methane production, and that surprised us a little bit, because normally what we see is that when NDF concentration or fiber concentration in a diet for the dairy cow increase, as it did here in this experiment, we normally also see a higher methane production, but this was not the case here, and that may have something to do with the improved fiber digestibility of the pulp to do. So that was a bit of the results, and we have considered a little bit how should we use pulp in the future for the ruminants. And if we look on the lactating cows, then we can say that generally it works fine when we substitute the grass clover silage with the pulp. But for high yielding cows, we expect that it only substitutes some of the grass clover silage, because we also see that it actually does decrease dry matter intake a little bit. You can say with the new political arrangements coming on, nitrogen leaching and so on now, then it seems like there's a huge potential for producing sugar beets, because nitrogen leaching is a little bit lower here. And I expect that this pulp would fit perfectly in a feed ration where if we start feeding more fodder beets. It's not something that we have looked into yet. Then we're also looking into other treatments of the pulp to see if we can increase the digestibility even more. And some of these treatments could, for example, be alkaline treatments of the pulp. But you can also say that that's for the lactating cows. We also have other animal ruminant groups, and that's among others heifers. It has not been studied much how heifers react on getting this pulp, but I expect that it would fit perfectly into a ration for them. Then we have also had an experiment running for dry cows, where it seems that we can replace the pulp. We can replace the maize silage that we use in rations close to parturition with some of this pulp instead. But there's something about the cation balance that you have to be aware of. Then relating inclusion of pulp in practice, then we have been testing it on some farms, as you see on the photo here. From Ausumgaard, there has been produced some pulp, and that was provided to some dairy farms, although it was not high-yielding dairy cows, but the perception of the farmer was that it worked quite well, and we can also see that the ensiling process and the quality of the forage or the pulp in these bales are also very good. And then for future, you can say what can the farmers expect? I think they can especially expect a lot of variation in the quality and the nutrient composition, and I think that relates a lot to what kind of setup they use. We will use in the future, if it's a local on-farm production of pulp versus a more centralized solution, where pulp will become sort of like a more bulk feedstuff. Then we also see there's a huge seasonal variation. Here is crude protein concentration shown for the different cut numbers here in Denmark, where we can see concentration is increasing a lot. I expect that that will also be reflected in the pulp that is produced throughout the year. And then there are some plant type species differences, which where you can say that biorefining and its effect on fiber digestibility will probably be better on some plant types or species compared to others. And then, as I said before, we may also reach an upper limit to how much the cows can eat, considering the fiber concentration. And I think maybe Christian will come back to this also later, but there will be a lot of logistic considerations also, making it work, how to put all this pulp into a bunker silo where it can ensile and so on, and get the like the right amount of pulp on a certain time so you can do this ensiling process. And then there are all the price relations that we may come back to, because right now it seems like most of the pulp, because of price, fits better into a biogas production. So conclusions from this is that we can see that the quality of the pulp varies as we know it from the grass clover silage. We can see that compared to the harvested crop, the crude protein concentration is generally either lower or quite similar, whereas the fiber concentration is higher. We can see, depending on the characteristics of the grass clover that we put into the biorefinery, this physical processing during biorefining, it can improve fiber digestibility, and it can also improve the protein value. And then I expect that for high-yielding cows, this fiber concentration, it may decrease dry matter intake, and that's why it's interesting to look on if we can combine it somehow with fodder beets in the future. And then final remark is that I think that pulp is an interesting feedstock for dry cows and heifers, and currently it's been tested for as a feedstock also for beef cattle. So that's my last slide. Thank you.
Thank you very much, Nikolai, for this good tour de force in ruminant feeding trials of the press cake fiber and pulp. Now we will, if you have questions to Nikolai to this presentation, you can ask them in the chat, yeah, I will not show the program slide in between because Christian is already on the way. That's good, Christian. So I will present the next speaker, who is Christian Knæ Drangsfeldt from Vestjyllands Andel. He's been working in the Danish feed company Vestjyllands Andel for the last years, involved in all the activity around setting up an actual commercial production of leaf protein concentrate from grass clover, and also utilizing the press cake fiber. So we'll hear more about how the industry can take up a technology and a concept like this. So Christian, please start when you're ready.
Thank you, and thank you for the invite to come here and talk to you all. Yes, my name is Christian Knæ Drangsfeldt. I'm working from a feed mill called Vestjyllands Andel, and I'm here to talk about the practical and the commercial experiences with biorefining during the last 3 to 4 years. Yes, and we are in collaboration with Ausumgaard. Have we built a, what should we call it, a pilot facility where we use biorefining clover grass and making the protein and making the pulp and making the brown juice. Yes, and I will talk a little about what we have been using it for and what we see in the future it can be used for, and which problems there have been, and all these things around doing this, and how hard it can be to go commercial with these things. Sorry. Yes. just a little some key figures of who are Vestinans Andel. We are a community. We are owned by the farmers. Right now we have like 4,125 Danish farmers. They're like our owners. There are around 250 employees. And we have 28 different departments around, mostly in the western part of Jutland. Our turnaround is 2.8 billion in 2001 and 2002. The profit was 114 million that year. And yes, and the profit have been that we have put back to our farmers since 2002 have been around 6 million. Yes, green development and Vestinans Andel. I have to move this. I can't see this. Yes. Yes, we have been in a lot of different projects, and we have always had it as our agenda to look into these new protein sources, because we can see that the soya we get from South America, or the organic soya from China or India, it's not robust always. We saw it very much when we had both the corona and when the ship was laying across the Suez Canal. All the prices was going up and down. So we need some more robustness to serve our farmers the best. So we have been looking into a different kind of locally produced proteins for many, many years. I think some of my colleagues started together with the AU for like 8 years ago, talking about this grass protein the first time and all the possibilities in it. Then we also have all this recirculation. We have a company where we try to pick up all the leftovers, we could call them, all the byproducts from companies that just work with grain and protein, different green proteins. And then we do a lot in green energy, especially from the biogas. And we're trying to create some synergies between all these different things. So we try to move our company and our not stakeholders, but all our farmers to be ready to the new world. We can call it that. Oops. Yes. Sorry. Yeah. I start out with we have the recirculation of waste products from the food industry, and then we try to make it to food or feed and biogas. So we have a company located in Billund. They are going around in the northern part of Europe. We're in Germany. We are in Denmark, of course, in the southern part of Sweden, and we collect all cookies, all flour, all these things, also some other oils and cake dough and all these things that are a little wrong. We also get a lot of muesli when there have been some kind of small mistake in maybe labeling it or something like that. So here we can take like a lot of products, and we have like these machines that can cut up and take the plastic off, or the pap off, or what else have been put into, and then we can use the products, and they don't have to go in the burning like they did earlier on. So this we make a lot of products from. We have something called cake mix. We use it a lot in our sow feed. We use it a lot to some of our in cattle feed, and we use it different places where we have need of energy, but also need of good taste, because this is very sweet and very good, and can get most animals to eat if we have some difficulties with that. And it's also done to reduce the feed waste generally. And it gives us a lot of good opportunities to learn other companies to know, so we get these synergies started. Yes. Then we have our own starfish factory located in Kostrup, up north in Skive, near Skive. It's close to Limfjord, and in the Limfjord, the starfish was quite a big problem because we had a lot of leaching of nitrogen and phosphorus and so on out to the Limfjord, and there was coming the mussels. The mussels. We tried to we have a lot of mussels that could clean up some of all these nutrients those coming in the water, and all these mussels, they were then being eaten by the starfish. And then there was a big project together with DTU that showed that that it would be okay to capture some of these starfish and then produce them into feed for animals. So we started that many years ago, and the mussel population, to improve the mussel population so we could get some cleaner water in the Limfjord. Yes, and this was done locally together with local fishermen. So when it was the mussel fishermen that could also collect these sea stars, and we're doing that for every year now for 4 or 5 years. Yes. And also up here, we are doing our fish meal, and we're doing some shrimp meal, and we're doing some mussel meal. And this is also for protein for animals production. Yes, then the green protein, and you know all the good things about it, but I would say it again. The green protein, we have a cover on the fields all year long. We have a high carbon capture. We're collecting a lot of nutrients. We don't need so much nutrients. It's also good for the organic farmer, especially to reduction of weeds for the next crop when you want to have after 4 to 6 years, when you have to have a new crop that can pick up all these nutrients that's going down there. And then it's a good way to produce a lot of biomass. And the biodiversity, we say it's higher, but I think we have not evidence on that so far. But we see if you have the right systems, you can do a lot in the biodiversity as well. Yes, facts of our raw material. We have the starfish 42 to 55%. We have the fish offcuts, and the fish offcuts is from the filleting industry. We use that to make our own fish meal, and we have the shrimps. And down there we have the grass protein, and it's like 75% to 61% of protein. Normally, you saw it also at Lene. She had a bigger variation, and this is in the dry matter. And also the fat content is between the 9% to 12% in the grass protein. And if we look at the soya, we have these 44 to 48% of protein. So it's a quite good protein, and the most important, especially in the organic feed, it's high on methionine, and we lack the methionine in almost all the other protein sources, and especially when we have, and we are good at doing that, we have faba beans and peas. There the methionine is quite low, so we need some feedstuff with high methionine, and there's grass very good. But grass is a little low on what's called cysteine, and that's not so good. But cysteine can be found in a lot of other products, so it's a good thing, especially for the pigs and for the poultry in general, on paper anyway. Yes, what have we used the grass protein for so far? Yeah, we have done it a lot of. I'm a pig nutrition, so we have done a lot of trials, what we call practical innovation. We have tried out different things on sows, castration sows. We have also tried it on lactating sows, and we have done it on small pigs, conventional and organic, and we have also done it on slaughter pigs up to, I think, 12% commercial in compound feed we have had. And the production results was good. We had hoped they will be even better, but so far they are just matching. But I don't think we are finished with the process of optimizing the protein. Horses, we make a lot of horse feed. Our brand is called Horse Lux, and we have 3 commercial sold feeds, and one is all-around feed, so people have the possibility to pick the green choice, we can call it that. And we have seen it also for horses to competition. People are very excited of it, and especially race horses are quite happy about getting this grass protein. They see that they perform better. We don't have any evidence, significant evidence, but some of them are starting to win, and they haven't done that before. That can be a lot of things, but right now they're saying it's because of the grass protein, and that's quite exciting. Then fish trials have also been, and some is ongoing. We also tried to feed calves with a few % of grass protein to see what happens here. And there have been a lot on poultry, but not so much with our protein. So we are right now together with KU starting to look at the poultry and grass protein in a commercial a trial with the poultry. And we have tried with the organic laying hens as well as it looked good. The problem have been that the manure, the feces get a little more sticky, but so far we have fed laying hens up to 6%, and we haven't seen in the stables we have looked into that was a problem. Then the next one is also pet food, especially dog food, feed, and maybe some cat feed as well. But that's the next step. Yes, here we have a story from one of the projects we have done. We took all the soya out and we used our own sources of protein, and we used fish meal and sea stars and shrimp meal, and we used the green protein. In the sow feed there was 5% green protein and 5% fish meal, and it was at the lactation and also the castration sows. In the small piglets we had the same mixture plus some extra protein from potato. And of course we have faba beans and peas in all the feed because we use a lot of that already. And the sorrel pigs was 7%, I think, grass protein, and the rest was horse faba beans and peas. And it had a solid production results using both the blue and the green protein, but we have hoped that it would be even better, but in this case it was just the same as you used to. Yes, but the blue protein was on standby because there was found some PFAS in the normal fish meal. So now it just have been okay again in organic production to use a little fish meal in the mixtures. Yes. Yes, all these things is okay, but we also have some experience from when we produce the feed. We can see the quality of the pellets is quite high when we use the grass protein. We had a trial with Aarhus University where they actually thought that the pellets, they were too hard, so we had to do something about that. So that also maybe especially to the small piglets and the weaning piglets that we have to consider, is it the pellet too hard for these pigs because the eating was not that good, and it was not taste, they concluded it was because they were too hard. So maybe the inclusion rate should be looked into. The other thing we have seen is it can reduce the frequency of stomach changes. We don't have facts on that yet, but it's something we're looking into all the time, and we're trying out, especially with these high-producing lactation sows, both conventional and castration sows and organic. But another interesting thing is that a lot of these high competition horses, they also have problems with the stomach ulcers. So maybe that's why they perform better, or the owners think they perform better when they get the grass protein. So we were looking for people that would like to look further into that. And another thing is that we have had with the pigs for a long time. We have organically, we have some problem with dysentery and Aarhus Biorefining, and we have used the grass protein with 4% in a herd that was a really bad case. And here they don't have a problem with the dysentery anymore. And they are still doing it here after 1.5 years, I think, and it has solved the problem. So that's quite interesting. So there's a lot of knowledge we still need to collect about this grass protein and all these things. Yes. Yeah. Just jump over this. Right now we're putting all the pulp almost that Nikolai don't get. We put that in the biogas, and we have 7 biogas plants around the western part of Jutland. And it's quite good and have been quite good in the biogas. So that's always a possibility. Yes, the biomass pulp can be used for cattle or biogas. The brown juice used for biogas or as fertilizer. We hope that the fertilizer would be a possibility, especially in the organic system. And the grass should be dried and used as feed product. And then we have the next step is taking the green juice and maybe trying to get out with the white protein, so we have something we can use for the humans. That'd be quite interesting and make a good case. Yes, future projects, grass protein introduced for large sections of the conventional pig production, and then develop new and improved grass protein plants. Right now, we're looking into 2. We were looking into 4, but all the projects didn't go. But now we're trying to start up 2 new ones, so it's quite interesting. But that's because the political climate in Denmark have changed a little. The white protein can change the whole business case of the grass protein. Yes, then I have just a little here. That's that. Thank you.
Thank you very much, Christian, for that really good presentation about all what you're doing, not only grass protein, but also to see that in a commercial setting, it needs to connect with a lot of other initiatives that you're also carrying out to produce local sourced protein, and grass protein will most likely not be the only one needed for that. So it's really good to see that you're testing and combining these approaches and getting also indications of something we in the university could be paying more attention to and make studies and research in depth of some of these interesting learning points you have from the feeding trials. So thank you very much. If you have questions for Christian, please post them in the chat, and we'll carry on the presentation line. So now we've been in Denmark and the Danish experience in this area, the last 3 presentations, and we'll turn to Ireland, where we have invited James Gaffey and Eleanor Scherer. James Gaffey is from the Circular Bioeconomy Group at Münster Technological University, and Eleanor Scherer is from Teagasc in Ireland. And you will present the experiences from green biorefining feeding trials in Ireland, and maybe put some words on the development that you see for green biorefinery there. So please, James, take over the presentation.
Thanks, Morten. Can you see my slides okay?
Yes, it looks perfect.
Perfect. Okay, thanks for the invitation. It's nice to be here and to share some of the results we're finding from Ireland. I'll start the presentation. I'm more on the technology side, but I'll talk about some of the trials we've been involved in, and then we've collaborated with Eleonora, who is more of a animal nutritionist background, and she'll continue some of the work she's led on developing. So so in terms of the potential for green biorefinery development in Ireland, I'm just going to kind of give a bit of context for why this is something we're really interested in, and also really interested in seeing what's happening over in Denmark, where I think in terms of fresh grass, fresh green biomass have been leading the way in the development of this area. From our perspective, grassland as a source of biomass, I think, is an enormous potential crop for bioeconomy development. We are the only country in Europe with over 50% of our total land area is grassland. It's far above other countries. We grow grass quite high yielding, and we have a lot of livestock production here in Ireland. So as a key development opportunity for unlocking potential, it's a big potential crop for developing new products. But there are other drivers. We have a big challenge around agricultural emissions, as a lot of countries do. We have over a third of our national greenhouse gas emissions which come from agriculture, which is the highest in Europe. So there's big pressure on the agricultural sector to reduce emissions. There are limits and reductions in place, targets in place at the policy level. Nationally, for reducing emissions by 2030, it's at 25%, which I know is lower than the targets in Denmark, but it's still a challenge for the sector here in Ireland to try and meet that change. Another area where we have a big challenge, and have had for a number of years, is the area of protein importation. And it's been mentioned in some of the previous presentations. Ireland, I think, is particularly susceptible in terms of protein imports. We import about 3 million tons of animal feed annually, which for a small country is a very large amount. We've had a declining cereal sector. We have a wet climate, so sometimes it's hard to achieve the productivity that we see in other countries, and we grow grass much more efficiently. But we need to find alternative protein concentrates then to meet the needs of our animal feed sectors, and a lot of that is coming from international sources. A lot of it is soybean, which is being imported, and we know that there are restrictions which are coming in place next year for the importation of soy for regions associated with deforestation, and that certainly, I think, is going to impact on supply chains here for all agriculture sectors, really, pigs, poultry, as well as cattle. And also we've seen this increase in cost, as a lot of countries will have seen over the last number of years in terms of agricultural inputs. It's approximately 73% over the last 7 years in Ireland. So this is something that I think, by better utilization of biomass, can create more resilience by using our local sources more sustainably, and maybe create some diversification opportunities as well. So we've done a number of projects in the area to develop this. The first project we did was a project called Biorefinery Gloss, which Nora and I collaborated on. And essentially, the focus of the project was trying to demonstrate a green biorefinery system on farm. It was a mobile system developed by the Dutch company called Grassa. And we collaborated with a number of partners on the academic side, MTU, where I'm based, and UCD, where Eleonora was based, as well as Carberry farmers, who are a dairy co-op, so they're the suppliers of grass, and working with Barry Row Co-op, who supply concentrate feeds to different sectors, including pig farmers. So we're trying to leverage this cooperative model of working with the grass farmers on the input side and working with the pig farmers on the output side. So I won't go through the green biorefinery process. People have already talked about this, but maybe just to say the products that we looked at were our press cake, which we looked at as a pulp, and Eleanor will go into that in more detail. And from the green juice, then we looked at the production of a number of co-products, the protein concentrate, obviously, which we'll deal with, and we've looked at only as a pig feed at this stage in Ireland, but we also looked at the production of this golden colored material called fructooligosaccharides from the brown juice fraction, which we tested as a potential prebiotic source for the promotion of healthy gut bacteria, probiotic bacteria in animal and human intestinal cells using some cell culture techniques. And we found very good results when we compared this to some on the market prebiotics in terms of growth promotion. And when we looked at this from an economic point of view, and I think it relates to the comment previously on the white protein, the production of this fructooligosaccharides product did a lot to enhance the economics, even if it was being produced in relatively small volumes. Okay, so I'm going to focus mainly on the pig trial work that we've done. So I think compared to Lene's presentations, our pig feed trials have been done at a higher level. They haven't been as detailed, and we haven't done as many of them. But we do have some first indications from the trials that we have done. They've mainly been conducted between us on the research side, but also collaborating with industry. So basically producing our protein concentrates, working with a local pig feed meal called Barry Rowe, who have done the rations and prepared the rations, and working with a pig farm in the region to look at how this pig feed or leaf protein concentrate could be integrated within weaner diets. And we found, I suppose, just to say the results, and then I'll show you some of the results. We found these are high level trials. We mainly focused on intake. Did the pigs like the protein feed, conversion efficiency, and weight gain? And overall, in terms of the findings, that we found generally positive potential for soybean displacement, which was the main focus of our study, and also a positive perception by the farmers and the mill and the industry partners that we've been working at. We didn't find any noticeable visual health impacts. We found changes like the change in slurry color. It's a much greener material. The concentrate itself, when in a ration, is a darker colored material, but we haven't found any health impacts so far visually in terms of what we've been looking at. So we've done 2 trials to date. The first was conducted in January and February 2021. It focused on leaf protein concentrate from perennial ryegrass. And we focused on a 27% replacement of soybean meal in weaner diets, 9-week-old weaners. And that's quite a low, I suppose, replacement rate. And part of the reason was that the grass protein concentrate that we were producing at the time was quite low in crude protein. So it was around 34%, whereas soybean is up around 44 to 48%. It was a little bit lower in some of the essential amino acid areas as well. And so we decided to be a bit cautious in terms of how we integrated this. And so we focused on this at a relatively low replacement rate. The trial duration was 31 days until slaughter. We had 55 pigs in the treatment and control diet. And in terms of our findings, we found, which we weren't sure what to expect really in terms of integration of the pig feed. The daily feed intake over the course of the treatment diet was about 6% higher than the control diet. So the pigs did like this grass protein in terms of consumption. We had a similar feed conversion ratio, and then the average daily weight gain for the treatment was about 6% higher than the control as well. So this work is being published. It was our first trial looking at it. That was conducted within the Biorefinery GLOS project. And then we were, I suppose, when we found these results, we were interested with the partners we had worked with to kind of see, was there an opportunity to go a little bit higher and see what the outcome would be for a more, let's say, consistent replacement of soya. So we've done some follow-on work in a project called Farm Zero C. Farm Zero C is a project which is trying to develop a climate neutral dairy model with the Carberry Cooperative in Ireland. And essentially, we use a commercial farm, which acts as a demonstration farm for integrating best practice in all kinds of areas around dairy sector mitigation. So we look at areas like animal emissions, with methane being a big problem for Ireland. It accounts for about half or nearly 60% of our total agricultural greenhouse gas emissions. So we look at things like methane abatement additives, slurry stabilization techniques. We look at best practice in animal breeding. We look at how we can improve soil and grassland, for example, through integration of clover, integration of multi-species swards, and other techniques. And we try and reduce the baseline emissions of the farm as much as possible, assessing this using life cycle assessment. But then we look at techniques we can use to try and offset some of the dairy farm emissions as well. So, for example, production of renewable energy on the farm, which can be sold to the grid. And then also what we're looking at more is this potential for green biorefinery as a way of doing this as well. So using our grasslands more sustainably and supplying products upstream to other sectors as well, while still producing milk, while still producing all the products we produce from milk. So within Farm Zero C then, we conducted our second trial, which was conducted during January and February 2022. This time the protein concentrate focused on a mixture of ryegrass and clover, and we increased the displacement rate to 50% of soybean meal with 9-week-old weaners. We had also an increase in the protein concentrate, so it's risen from around 34% to 43%, so it is more in line with what we would expect soybean to be, although lower than some of the work that we're seeing coming out of Aarhus University. So we decided to increase this to 50% replacement. The trial duration was 31 days until slaughter, and we doubled the amount of pigs in the treatment and control. So it was 110 pigs. So after kind of initial acclimatization, the daily feed intake of the LPC diet was 2% higher than the control diet by the time we got to the end of the trial. Not much of a difference in terms of feed conversion efficiency, and by the end of the trial we had a slight increase in average daily gain by the end of the trial for weaners on the leaf protein concentrate diet or the grass protein concentrate diet. So again, we found this is a positive performance. We're keen to go a little bit higher again in terms of the displacement rate, and to try and improve as well in terms of the actual crude protein and amino acid composition. Again, in the amino acid composition, it was improved versus the first trial, but we were still a little bit lower in terms of lysine and cysteine in the amino acid side. But we do feel like there's good potential there to develop a new industry around this for Irish grassland, especially given the potential, of current grassland situation in Ireland. I'm just going to add, and Eleanor is going to go into the ruminant work in more detail, but we did, as part of the Farm Zero C trial, have a recent press cake ruminant trial, which was conducted by Hazel Costigan, and the publication has been accepted, but we're still awaiting for it to appear online. This trial was done during December 2021 to January 2022. It was alongside the second feed trial that we did. So you can imagine part of the protein concentrate going to feed the pigs, and then this was the press cake that was going to feed the cows. And we replaced 60% of the silage within a conventional dairy diet with press cake that we produced in the biorefinery with the same amount of concentrates in both diets. The study was undertaken during late lactation, and the study particularly focused on methane production, which, as I mentioned, is a big challenge nationally for us in Ireland. Eleanor, I think, will go into more detail because she's also looked at methane as well as a number of other parameters in her dairy trials. And we used the green feed emissions monitoring system to conduct the trial, and we had 21 cows in each trial. So what we found was, in terms of methane production, we found in the case of the press cake cows, we had a 6.3% higher production of methane for the press cake cows during late lactation. But we also found that the pressed cake cows produced nearly 10% higher fat and protein corrected milk. So the methane then is primarily being driven by higher group average dry matter intake for pressed cake cows of about 3.6% higher. And we find then that there is actually lower emissions of methane on a fat and protein corrected milk basis. So it depends really then on whether you're looking at this on a per animal basis or a per fat and protein corrected milk basis. Overall, from Hazel's side, the findings would indicate that pressed cake diet increased absolute methane and milk production, but when methane was expressed per kilogram of milk and dry matter intake, there was no major difference between the treatment and control. I know some of this is Eleanor is going to look at in a little bit more detail through some other trial work at this as well. So the next stage for us in Ireland, we have received funding for developing pilot and demonstration scale facilities to advance the work in green biorefinery from the Department of Agriculture. In the case of the demonstration plant, this is going to be based at Farm 0C, so it's actually going to be built onto the farm, and it's going to be put in place with an anaerobic digestion unit as well. And then the pilot plant will be based at Munster Technological University. And I would say this is something we're excited about developing. And in the case of the work we are doing, we are getting significant collaboration from the team at CBIO in terms of this development, and learning from the experiences there has been extremely helpful, and a lot of support from CBIO in terms of the work we're doing. And just to mention some of the future research activities that are coming up on higher value additives in animal nutrition. So we have just commenced a project funded under the Circular Bio-Based Europe joint undertaking called Rural Biorefineries, which will be a demonstration scale project. And it includes collaboration with stakeholders from all over Europe, but there's a big Irish-Danish collaboration, including the team at CBIO and Aarhus in the project. And this project is going to explore new possibilities for nutritional products and additives. So where it was mentioned about the production, for example, of food-grade protein, we're going to be looking at all the current streams that are produced in a green biorefinery and trying to understand what's the potential for developing and scaling value-added products, including production of white protein, which the team at CBIO and Aarhus are going to be leading on. We're also going to be looking at the production of volatile fatty acids and medium chain fatty acids from our anaerobic digestion process using the brown juice fraction of the green biorefinery. There's international studies which are looking at the potential of medium chain fatty acids as feed additives in pig production. So some of the work we're going to be doing will be assessing the bioactive role of these fatty acids in nutritional management of pig gut health, and potentially the antiviral capability against specific viral pathogens, so things like PEDV, which impacts on young pigs. And this could be interesting because you have pig farmers, for example, faced with trying to deal with the reducing role of zinc oxide as an additive in piglets. So there is potential maybe for certain products like the fatty acids from grass, or even the fructooligosaccharides from grass, to improve nutritional health in young pigs. But this is something that we hope to investigate, and we plan to investigate using in vitro and in vivo work using the new project that's coming up. So I think that's my slides. So I'm going to pass over to Eleanor, who's going to go into the press cake work in more detail.
Yeah, thank you, James. I'm going to share now. Can you see my slides?
Yep.
Yep. And okay, so thank you very much. My name is Eleonora, and I'm presenting the results of my PhD program, where I looked at the potential of the biorefinery press cake as feed source for dairy cows and its potential of reducing nitrogen and phosphorus excretion in a dairy system. During my PhD, I work with mainly two projects: the biorefinery glass that looked at press cake made from fresh grass, and the Live Farm for More project that looked at press cake made from sustainable produced silage.
Eleanor, your slide is shown not in presentation mode.
Oh, sorry. I'll try again. Sorry.
No worries. We have time.
Can you see in presentation mode now?
No, it looks like you're pressing the presentation mode button, but it stays in the other.
I
Are
could
you
share
operating
your
two
slides
screens?
if you want, Eleonora.
Yes, please, James.
Okay, 1 second.
So in the meantime, I can mention if you have questions to James also, please write it in the chat. And James, if you can have a look also.
I think I've only got the PDF version, so I'm not sure if it's any better. Okay, I can try anyway. See. Yeah.
That's perfect.
Okay.
So sorry. So as I was saying, I collaborate with two projects. One looking at the fresh grass as starting biomass with the biorefinery glass, and the Live Farm for More project looking at the press cake made from sustainable produced silage. So looking at the press cake made from grass, fresh grass, I ran two experiments: an animal feeding study using 30 autumn calf in early lactation Holstein Friesian dairy cows that at the start of the experiment were on an average of 70 days in milk and were producing an average of 31 kilos of milk per day at 4.3% fat and 3.7% protein. Cows were divided into two groups and were assigned one of the two dietary treatments, grass silage only or press cake, where 66% of the forage was replaced with biorefined press cake silage. The experiment ran for 77 days, with 14 days of acclimatization period and 63 days of experimental period, during which measurements were collected for daily dry matter intake, milk production and composition, rumen fermentation parameter, metabolic status, and for nitrogen and phosphorus balance. Same diets were then used in an in vitro study using the rumen simulation technique for an 18 days of incubation period with 10 days of acclimatization and 8 days of experimental period where measurements were taken for in vitro apparent digestibility, in vitro fermentation parameters, and total gas emission production. The main difference between the two treatments were on higher dry matter intake, on higher dry matter content and higher fiber content, and a lower protein content for the press cake diet compared to the grass silage only diet. As I said, 66% of the forage was replaced with biorefined press cake silage. But animals received the same amount of concentrate. So diets were formulated to meet animals' nutrient and energy requirements, but were not balanced for any nutritional values, as you can see, because the main objective was to see what happens if we replace silage with press cake silage. So the main findings were a reduction of dry matter intake, a reduction in rumen ammonia concentration, an increase in nitrogen excretion in milk, a reduction in nitrogen and phosphorus excretion, and an increase in nitrogen use efficiency for the cows that were offered the press cake silage, press cake diet compared to the grass only diet. Then from the in vitro study, we have seen a reduction in dry matter disappearance with the press cake diet compared to the grass silage diet, and these come in contrast with the findings of the animal study, where cows, where the diet seems to be more digestible in cows, and that could be due to the system, as the rumen simulation technique, as you see, it's only mimic the rumen side of the digestion, so it could be that we are missing part of the digestibility there. But rumen ammonia concentration was lower for the press cake diet in the in vitro study as well. And just to add on the methane results, we didn't see any significant difference between the grass silage only and the press cake diet in the in vitro experiment. Moving on my PhD, I collaborate with the Life Far From More project that looked at press cake made from sustainable produce grass clover silage. Sustainable produce means that it wasn't an organic system, but it was produced without any inorganic fertilizer. So only slurry was applied to the grass clover mixture. I won't be going into too much detail of these results as they're still under publication. I conduct in 2022 an animal feeding study using 6 autumn calving late lactation Holstein Friesian dairy cows. They were at a later stage of lactation compared to the first trial, at an average of 185 days in milk. This time we looked at 3 different inclusion replacement levels of press cake silage. So we had a 0% or only grass clover silage, a 25% replacement with grass clover press cake silage, and a 50% replacement. The trial ran for 3 periods in a crossover design with 13 days of acclimatization period and 9 days of experimental period, where measurements were collected for daily dry matter intake, milk production and composition, rumen fermentation parameter, and nitrogen and phosphorus balance. This time, having a smaller number of cows, we were able to collect feces for the nitrogen and phosphorus balance, feces and urine separated, and 24 hours. The main findings were, again, a reduction in dry matter intake for the PC50 diet compared to the grass silage only diet. Again, a similar milk yield with a reduction in milk urea, an increase in the proportion of nitrogen excretion in milk for the PC50 diet compared to the other two treatments, with an increase in nitrogen use efficiency and phosphorus use efficiency, and a reduction in total nitrogen output as kilos per day in PC50 compared to PC25 and and grass silage. So, to conclude, from my experience using press cake silage, we can say that press cake silage can act as a partial replacement for traditional silage in the diet of lactation dairy cows without impacting milk yields and milk quality. And from the reduction in nitrogen and phosphorus excretion and the increase in nitrogen use efficiency and phosphorus use efficiency, this indicates that press cake silage has the potential to can have a beneficial effect on the environment when used in the diet of dairy cows. Thank you very much.
Thank you very much, Eleanor, for a very good presentation and a detailed tour in the experiments on the ruminal animal trials you have done in Ireland. So that concludes the presentations, and we still have 12 minutes left for a few questions and discussions. Please post in the chat if you have further questions, and otherwise I will moderate. I hope all previous presenters are still with us. Because I would like to draw a few things. So from Lene, we heard the experiments, this experience on the monogastric animal feeding trials and how processing and the feedstock really impacts the quality and the digestibility of the products. And Lene, I would like to maybe ask you, how do you see sort of going forward with the experience you've seen in the for the protein concentrate feeding trials? How do you see us going forward? What should we focus on in the processing part and in the field to gain the most value and the most relevant product for the monogastric animals?
I think we have to be aware that it is a different material we're working with compared to the traditional protein sources. So it's you know, a functioning biomass that we work with. It does not necessarily mean that we can't use it, but we just have to look at it in a different way than, for example, soy or other dried protein sources. So in the production chain, it is something that we have to manage this all these processes that will happen when we as soon as we harvest the plant. And as I said, when we try to optimize the overall biorefining process, we need to focus on all these different aspects. So we can do something to improve the protein content, but if that's at the expense of the quality, the overall benefit is maybe not that good. So perhaps we have to acknowledge that, you know, from a technological point of view, we can increase the protein content, but we will not gain anything on the quality. it's kind of we have to look at this balance, and this needs to be a focus in the future. So I think it's it's not easy, but it's manageable. But we have to be aware of what we're working with.
Yes. Thank you for that. Also, I would like to include some of the questions that have not been answered, or maybe could also be elaborated. That's in the chat. And with regards to you, Lene, did you just post a question here? Have there been trials using the protein juice in wet pig feed?
We haven't done anything in Denmark, but I think they have done some in Norway, where they have treated the juice with the acid to increase the shelf life of it, and then included it in the feed for the pigs. And as far as I know, it has been successful. You could say that the issue with using the protein juice compared to using the dry protein concentrate is that you have an active product, so you can't just store it as it is. But I can see that it is possible to use it, but again, you have to handle it in a different way than the dried concentrate.
Yeah, I think also there's been, or I know there's been trials in Sweden as well with juice produced from silage.
Yeah.
Yeah. So the amino acids and the peptides there will also have a function as a feed potentially, but an area I think we need to look more into if we're going forward. I would like to change to the press cake part, and Nikolai, you in your presentation you also mentioned several times a few comments about. It looks as it really can have some benefits in ruminal nutrition, and in combination with other things going forward, also introducing new types or maybe old types of feed with the sugar beet or fodder beets. But my question is more, what is needed if we are going forward to use much more of the press cake for ruminant feeds compared to biogas? What is important in the value chain to focus on to make the best possible ruminant feed?
Good question, Morten. I think, like in general, it is actually quite good ruminant feed already, and I would just like to highlight a project that we are participating in also together with Ambye-Jensen called Climate Reach, where you can say we can also together with the field aspect improve on the overall scale our climate impact. So here we want to introduce some new forage species, grasses, not new forage species, but we want to introduce more, for example, tall fescue in the diets for the cows, and that's not necessarily good because we normally also see lower digestibility and so on. But it seems like based on the digestibility trial, we see here that if we just improve the fiber digestibility by this processing, then it seems to be better. So I think there's a lot of, we need to put more focus on all these on the kinetics in the rumen, fiber digestion, fiber passage, and so on, to see if we can like improve the digestibility even more in different treatments, through different treatments, or combining with other species like tall fescue. And one of the things for now is to include this alkaline treatment, which hopefully opens up more of the particle or cell wall structure and releases more protein in the meantime, but also improve the digestibility of the fiber. So I hope that answers part of your question.
It's probably a big open question, a lot of relevant, but yes, very relevant. And I'd like to turn it to you, Christian, staying on the use of the press cake. This was also a question posed in the chat, which you answer, but there's, I think, this issue of what to use the press cake for. Now it's biogas, and as you write, that is the easy available solution on site in many aspects. What is your take on the development further for if we are to put the press cake fiber for ruminant feed instead of biogas? What needs to be developed from where you're sitting?
Actually, not that much, because if you can use the press cake as it is today, and we're using doing it, the process is optimizing all the time, so there will be a little less protein for each year, depending on how the season looks and all these things. And we can see it varies a lot, but the cattle people are used to that. We saw Nikolai's first drawing of how it was in the different cuts. So they're used to optimize for that. So I don't think it's probably, I actually think it's already ready for the cattle people. And we can see when we store it in big silo storage, it's not a problem. It's good also when we take it out and put it in the biogas, so it can go other places. Right now it's a value, and I don't think the farmer is ready to shift it with the grass they have at home so far, and we also have the logistic in it to move it out to the farmers and so on. But I think the product is ready, and people are welcome to come by it.
That's good to hear. And are there any of the welcomed people that actually ask for it? And so the farmers, the dairy farmers, or farmers with ruminants that asks for this product, or is that not something?
No, we have sent something to a farm nearby, Ausumgård, where they have tried it. I don't know how many loads they have got, but they have tried it in a period. But I haven't got the data. But I can get Christian to send it for Nikolai if he wasn't involved with it. I'm not sure. But we have also tried the fiber for sows out on a farm, organic farm, and it also looked quite good. And we tried it for slaughter pigs, and they're not, they don't like it as much as fresh grass, of course, because the taste and all these things are lower. But we also have, when we are better and better to pressing out the protein, all these things, we maybe can use it for something else as well. And I know you are also working with that. So I think we have to look at the whole picture. Where's the most value of the project? In this time, and maybe in a few years, it will be something else. As the same with the protein. Right now, it's for animals, but maybe we can clean it up and use it for something better. Also for humans, like the white protein, or maybe people are getting adjusted to the taste of grass.
Yes. There are a lot of possibilities. I fully agree, but it's good to hear that it actually works well and can be put in a silo and ready for also the ruminant feed. I'll maybe use a few minutes. I want to just turn to you, James, in Ireland, and I'm really happy to see the developments there, and looking forward to collaborate on this new project in CBIO with your pilot facility. There was a question in the chat around carbon footprint that Lene asked, but I know you have also been looking into carbon footprint of the green biorefinery systems and the leaf protein concentrate. Can you put some words on that, and because you mentioned in Ireland that is also a big focus and for your Zero Farm C project.
So yeah, and we've actually collaborated with a paper with Morten and Taras in CBIO. So we were looking at the potential footprint of grass-based protein concentrate, assuming different feedstocks, one looking at ryegrass and one looking at a clover grass mixture, and then looking at different processing pathways for producing the protein concentrate and comparing this with the imported soy. And I guess we found that we could increase the footprint of the protein concentrate, producing it from protein from green biomass by about 5 times lower of a footprint compared with soybean that would be imported, a kind of international mix, but mainly imported from South America. So there is some really nice potential there. But we also have a challenge in that at the moment, in terms of how how the national inventory is calculated here, we assume that the footprint of protein is it's burdened with the production company rather than a country rather than the use country. So this is kind of a challenge because at the moment it's allocated at the source of production rather than the use. So in the case of Ireland, it could be something that's on paper a more sustainable exercise to import protein than it is to actually produce our own protein, which I know doesn't make very much sense, but it's something that's on paper that's something that is a challenge. So even if we can produce let's say protein more sustainably from local resources, at times we're not getting the credit for making those decisions.
Yes, yeah, and this is also what we see in Denmark. I mean, the business case is challenged very much, and but the good arguments for the system's environmental arguments are so many and a lot, and they are not valorized as at the moment. So I agree on that. I think we are 3 minutes over time, so I'll not take more of your lunch break. This is, I mean, short 2-hour webinars. We cannot cover everything, but if the topic caught your interest, please get in contact with us or follow the development around the Danish Green Biorefinery, but also the Green Biorefinery activities in Ireland and other places in the EU. It seems like there is a momentum at the moment, so we're really happy to push this as far as it needs to be an actual impactful technology for agriculture. So the recording will be posted on our homepage, and hopefully we'll see you next time for a CBIO webinar. So have a nice day, everyone.
You too. Bye bye.
And thank you for the speakers.
Thank you.